Welcome back, focused learners. Today, we're diving into a highly concentrated explainer, basically a master's study guide designed specifically to help you conquer some of the absolute ultimate medical exam villains. We're talking about the non-fermenting gram-negative bacilli. If you're looking to lock down those high-yield microbiology facts, well, you're in exactly the right place. But hey, before we get into the heavy science, let me ask you a quick question. What connects green nails, a relaxing hot tub, and a hospital ventilator? Sounds like a weird riddle, right? But the answer actually lies in a very specific group of microscopic survivors. Keep that in the back of your mind as we start activating your clinical pattern recognition. Okay, let's dive into this. First, we'll meet the villains broadly, and then we'll break down the big three: Pseudomonas, Burkholderia, and Stenotrophomonas. After that, we're going to build up to our ultimate organism comparison guide using some high-yield tables. And finally, we'll wrap the whole thing up with those essential exam warnings you literally cannot afford to miss. All right, Section 1, meet the gram-negative villains. So in the microbial world, these guys aren't your typical everyday pathogens. They're notorious opportunists. By definition, non-fermenting gram-negative bacilli are bacteria that simply do not ferment carbohydrates. Unlike the typical enterobacterials that are just hanging out, living harmlessly in your GI tract, these bugs are mostly environmental survivors. You find them in soil, water, moist environments. And because they aren't part of normal human flora, they generally need a moment of weakness, a compromised host, to cause a true infection. But man, when they finally do strike, they are incredibly patient, and they notoriously resist our standard antibiotics. Moving right along to Section 2, Pseudomonas, the opportunistic boss. If these bugs were an organized crime syndicate, Pseudomonas aeruginosa is undeniably the big boss. Looking at its foundational microbiology, it's a gram-negative, non-lactose fermenting modal rod. Uniquely, it thrives and actually grows at 42 degrees Celsius. But listen, if there is one single thing you must permanently burn into your brain right now, it's that this bug is oxidase positive. That is a massive, game-changing differentiator on test day when you're staring at lab results. And you'll often recognize its Pseudomonas without even looking at a chemical lab report, just because of its vivid sensory clues. It produces this distinct yellow-green or blue-green pigment called pyocyanin, and that literally causes green discoloration of the skin, of wounds, or even nails, which actually answers the first part of our opening riddle. When it's cultured, it forms these shiny gunmetal colonies and emits a really distinct sweet, fruity, or grape-like odor. These sensory details, they are absolute goldmine memory triggers for your exams. And this brilliantly illustrates two of the most critical clinical syndromes associated with Pseudomonas, which are deeply tied to very specific patient profiles. Take ecthyma gangrenosum, for example. Think of profoundly neutropenic patients or those with AIDS. It presents as painless maculopapular lesions that morph into purple, then black necrotic skin lesions, all due to vascular invasion. Contrast that with malignant otitis externa. This is almost exclusively seen in elderly diabetics. They present with severe ear pain, sometimes a cranial nerve 7 palsy, and an incredibly high ESR of 100 or more. Totally different vulnerable patients, but the exact same boss-level bug. Let's keep the energy high with a rapid-fire rundown of other classic Pseudomonas associations. It's a huge cause of ventilator-associated pneumonia, which, you guessed it, links right back to the ventilator in our opening riddle. It causes hot tub folliculitis, a self-limiting rash after soaking in contaminated spas. Then there's keratitis, an absolute medical emergency that can literally perforate the cornea for contact lens users. Oh, and puncture wound osteomyelitis? That's famously seen when a kid steps on a nail right through the rubber sole of their sneaker. So how exactly does it pull all this off? Well, it has a vicious type 3 secretion system that injects cytotoxins directly into host cells. Strains that secrete the XOU toxin, those are linked to significantly higher mortality. Plus, it produces alginate, which is a mucoid exopolysaccharide. This forms a really thick, protective slime layer that shields the bacteria from phagocytosis, and that leads to the hallmark mucoid strains that chronically infect patients with cystic fibrosis. Section 3. Burkholderia, tropical threats, and cystic fibrosis. Now, this genus is basically the deadly cousin of the Pseudomonas family, and examiners absolutely love testing its geographical and occupational traps. Let's compare two massive threats side by side. First up is molioidosis, caused by Burkholderia pseudomali. This one is environmental, found in soil and water, and heavily linked to Southeast Asia and Northern Australia. It often presents with an upper lobe pneumonia that brilliantly mimics TB, complete with cavities. On the flip side is glanders, caused by Burkholderia mali. The critical difference here? It is not environmental. It's strictly an occupational zoonosis linked to horses and equine handlers. Then we have the Burkholderia sapata complex, and I really want to emphasize how incredibly serious this group is. In patients with cystic fibrosis, this can cause Sapasha syndrome, presenting as rapidly fatal respiratory distress and septicemia. It's so devastating and intrinsically resistant to so many drugs that certain strains, like B. Stenotrophomonas, are actually considered a major barrier to lung transplantation, simply due to the extraordinarily high post-transplant mortality.
On to Section 4, Stenotrophomonas, the resistant survivor.
So if Pseudomonas is the big boss, Stenotrophomonas multophilia is the apocalyptic survivor, just thriving in a microbiological wasteland. It is a strict opportunist. It essentially doesn't cause problems until we, in the hospital, inadvertently create the perfect environment for it. It emerges specifically when a patient is given very broad-spectrum antibiotics, like advanced cephalosporins or carbapenems. Those heavy hitters eliminate all the normal competing flora, leaving a totally vacant microbiome wasteland for this highly resistant scavenger to just rapidly take over.
So, the crucial point is how you actually fight it. Stenotrophomonas is inherently resistant to carbapenems because it produces specific L1 and L2 beta-lactamases. If you try to treat it with your biggest, strongest hospital antibiotics, you will fail. Its true kryptonite, actually, it's the classic older antibiotic TMP-SMX. That is unequivocally the drug of choice for this particular survivor. All right, Section 5, the Ultimate Organism Comparison Guide. We've met the villains, so let's build out our master cheat sheet tables to really lock this knowledge in. Walking through this first table, looking at key features and pathogen types, Pseudomonas is your oxidase-positive green pigmented opportunist. But look closely at the Burkholderia species, Pseudomali and Malay. Notice something different? They are explicitly listed as primary pathogens. That means they don't just wait around for a compromised host, they're actually capable of infecting a healthy person outright. That's a super vital distinction to remember.
Building on that, this next table visually maps the organisms directly to their classic clinical diseases and primary risk factors. Pseudomonas pairs up strongly with neutropenia, cystic fibrosis, and diabetes. Burkholderia sapasha is practically synonymous with cystic fibrosis and chronic granulomatous disease, and our survivor, Stenotrophomonas, pairs tightly with a history of prior broad-spectrum antibiotics.
We'll conclude our matrix with the treatment cheat sheet. I highly recommend you literally take a mental screenshot of this matrix right now. P. aeruginosa requires an anti-pseudomonal beta-lactam, stepping up to drugs like ceftolozine tazobactam for multi-drug-resistant strains. B. pseudomolii needs an intense initial IV phase, followed by a grueling 12-week or longer eradication phase with oral TMP-SMX. And again, Stenotrophomonas is uniquely treated first-line with TMP-SMX because, as we know, it inherently resists carbapenems. Finally, Section 6, High-Yield Exam Warnings. Examiners absolutely love setting traps, so let's get a bit conspiratorial here and cover the rapid-fire exceptions. Hear this loud and clear. Warning: Stenotrophomonas is intrinsically carbapenem-resistant. Do not let yourself forget this fact. Hitting this bug with a carbapenem on your exam or in the hospital is a massive clinical error. It's the exact class of drug that probably caused the opportunistic overgrowth in the first place. And here is another major pitfall to watch out for. If you see ecthema gangrenosum in a neutropenic patient or malignant otitis externa in an elderly diabetic, your brain should immediately flash to Pseudomonas. Now, technically, in the real world, these lesions aren't exclusively caused by Pseudomonas, but on test day, board examiners use these exact specific patient pairings as giant flashing neon signs pointing you straight to the correct answer. Let's end with one final active recall test to see if you're ready. If a veteran returning from Southeast Asia presents to your clinic with TB-like cavities in their lungs, what latent non-fermenting bug should you immediately suspect? I'll let you review your master tables for the answer to that one. Keep these high-yield associations locked in, and you will absolutely crush test day. Thanks for joining me on this explainer, and happy studying!